humble tubes parsing nutrients and waste from mouth to anus. Yeah, that's kind of the plodding existence that we all lead. All right, so some of the history. For a long time, the GI tract was not really embraced as a worthy academic endeavor, a worthy endeavor for research. Because you eat food, tastes good, you poop. Not especially exciting to people. But here, I'm going to tell you about a couple fun things from it. So I love this. It may not seem humorous to any of you, but it really does to me. Da Vinci said the digestive system aided the respiratory system in its function. Okay, so follow that for a second. The digestive system aided the respiratory system in its function. What is he getting at? The compressed intestines with the condensed air which is generated in them thrust the diaphragm upwards. The diaphragm compresses the lungs and expresses the air. Yeah, so what he's talking about here is flatulence. Da Vinci clearly had a little bit of gas, some compressed air in the intestines. I think that's pretty funny. Da Vinci come to terms with his farts. The stomach is the lowest and has a hidden place in the body because of its uncleanness. So this is getting at this notion that people don't really want to talk about the stomach. The ancient Ayurvedic teachings held that there are these chakra that run up the body, and the lowest of them is your basest instincts, the lowest expression of your humanity, whereas the higher, the further away from that you get. Yes?
This related to Galen Hippocrates about
Absolutely. So I only have so many slides, but yes, this is like a direct inheritor, the imprint of Galen and his nemesis. So let's finish out Benedetti's quote here. The stomach is lowest and has a hidden place in the body because of its uncleanness, as though nature had spared the principal members and had relegated the stomach or bowels farther away from the sight of reason and of the mind, and fenced it off with the diaphragm in order to not disturb the rational part of the mind with its importunity. These members serve the higher ones. Some of them concoct the food into juice, others digest it into various humors, and others expel the superfluity. Alessandro. It wasn't actually until a guy named William Beaumont. Has anyone heard of William Beaumont before? I was born in Beaumont Hospital in Detroit. Didn't know anything about him at the time, but now I can tell you the story of William Beaumont. He's an interesting character that was a field surgeon during the French and Indian War that saw one of these soldiers, one of these frontiersmen, take a bullet to the gut, not actually on the front lines. It was a accidental discharge. And Beaumont, turns out, was not actually a very good surgeon. And so he went in there, he's like, oh my God, there's a lot of blood, I gotta stop it, let's sew stuff together. And he sewed some stuff together, and then put a Band-Aid over it, don't look at it, and hope that his next of kin are at hand for his imminent demise. But it turns out these frontiersmen were made of some pretty sturdy stuff, and this guy didn't die, and in fact went on to survive quite well. But it was revealed that Beaumont made the mistake of sewing the outer edge of his body wall to the inner lip of his stomach. So this guy literally had a little window, a little flap you could open up and look inside of his stomach. And Beaumont he was like, that's incredible. And started taking bits of food, tying them to strings, opening it up and sticking in there and closing the hatch and letting it digest for a while, and then like open it up, yank the string and pull it out and see what he had there. And he began to like take copious notes and develop this huge archive of descriptions of how different foods were partially digested by the stomach. And this was the beginning of the real science, our scientific understanding behind digestion, by the foibles and curiosity of a sort of hack field surgeon. I could make a slide for that, but you heard the story nonetheless. So here's an overview. What does the GI system do? Well, it's going to process and digest food, of course. We all know that. But it's also important in the absorption and conservation of water. It's going to, this food that we have digested, it's going to absorb the relevant nutrients. These are, of course, ions and water, but then the breakdown products of the three primary macronutrients, the macronutrients, right? So macronutrients are proteins, carbohydrates, and fats. And then also absorb whatever micronutrients, meaning vitamins, cofactors, minerals, stuff like that. And then lastly, it will help to store energy reserves, specifically in the liver, in the form of glycogen. We'll get there. Got an hour. Let's see how far I get. So I've already talked about this, 5 chemical families of taste. So I'm not going to belabor that. And I've talked about labeled line code as well, and how these labeled line codes are mapped onto the gustatory cortex in the brain. We've talked about that quite a bit, so I won't double up on that for the sake of time here. But I am going to talk about this. That this is kind of an interesting thing. We have 2 separate taste circuits, afferent, efferent circuits. Two separate taste and salivatory circuits. Taste being the afferent side of that, and salivation the efferent, the motor output part of it. And they directly correspond to the anterior and posterior surface of the tongue, or the anterior and posterior regions of the oral cavity. So the anterior tongue, the taste is conveyed along cranial nerve 7, the facial nerve. And the facial nerve takes the info up to the gustatory cortex. Whereas the anterior portion of the tongue is separated by the lingual sulcus from this posterior half of the tongue. And if you look in the mirror, stick your tongue way out, look in the mirror, you'll be able to see that there is a real difference in the texture between the front and the back of your tongue. So the taste back here is coming from cranial nerve 9, the glossopharyngeal nerve. The glossopharyngeal nerve. So the taste in the back of the tongue and in the oral pharynx. We actually have taste receptors in your oral pharynx as well. But both of them bring it to the gustatory cortex, and then each of those nerves brings efferent innervation out. So the parotid gland receives its efferent input from the glossopharyngeal nerve, whereas the sublingual and submandibular glands that are more anterior, bringing saliva into the front of the mouth, are served by the facial nerve. All right. And I had mentioned that these different glands have different types of saliva. The parotid gland makes the most watery of the saliva, very watery. Produces a serous, watery secretion. Whereas the sublingual gland down here is going to produce a saliva that's very mucinous, or has a lot of mucus in it. It's mucins are these glycoproteins. And then the submandibular gland is actually somewhere in between. It has a mix of the serous and the mucus secretions. So I find that to be kind of interesting, that there's two totally separate afferent-efferent loops for taste. All right, so what's happening in your saliva? Saliva has this protein called salivary amylase. It's also sometimes called ptyalin. PTY, who doesn't like that? Blend. PTYALIN, ptyalin. Hydrolyzes the alpha-1 for glycosidic linkage. So a glycosidic linkage is simply the linkage that connects two different sugar rings, right? So in starch, animal starch, it's also called amylose, the sugars, the glucose, are all linked up to one another with alpha-1,4 glycosidic linkages. And the salivary amylase begins the job of cleaving off some of the sugars. All right. So here's how all of this gets regulated. Here are the different layers. This is the anatomy that we talked about in the lab today. Serosa muscularis layer, submucosa, and mucosa. But there are three ways that this is controlled. There's a neural loop. It's this long reflex where we have stretch receptors and chemoreceptors in the muscularis mucosa that send a signal up to the central nervous system that sends inputs into the myenteric plexus. So there's both long and short paths. And when that happens, we start to get this peristalsis, these waves of muscular contraction. The second is hormonal. So there are various secretory cells that are going to release a host of acids and buffers and different enzymes. And there are going to be hormones that are going to be released by these same cells into the bloodstream that are going to circulate and hit via the bloodstream, hit other regions in the GI tract, turning on what are called the enteroendocrine cells. There are going to be local factors. These acids that are going to be released are going to change the pH, the physical stimulation of having food there, the chemical stimulation of maybe some amino acids or some fat or what have you. That's going to stimulate direct local effects that are going to have these secretory cells accelerate their activities. All right, so we did the gastric anatomy already. I'm just going to remind you, we have the gastric pit that leads into the gastric glands, where we have these chief cells, the parietal cells on the walls of the gastric gland, and then there are these G cells at the bottom. I'm going to talk about each of those cells and what they do right now. All right, first, in these gastric, so here's a picture of the gastric pits. Emerging from these gastric pits is going to be hydrochloric acid. And hydrochloric acid can have a pH that's as low, it's below 1, so like 0.8. That's extremely acidic. That's really, really acidic. How do we do that? How do we make that stuff? Well, it's the job of the parietal cells. So this is how this works. We have carbon dioxide in the cells, which gets combined with from a metabolic byproduct of carbon dioxide, combines with water via an enzyme called carbonic anhydrase, which is going to make this carbonic acid, which dissociates into bicarbonate and the conjugate base of this acid is bicarbonate and the hydrogen ion. So this is acidic, right? And we're going to take this bicarbonate and we're going to exchange it, keeping a charge neutrality. We're going to use an antiporter to exchange bicarbonate and bring in chloride. All right, so we're trading anion for anion. This is called the alkaline tide because this is basic, right? And this helps to buffer the blood. This makes your blood more alkaline. And then this chloride then gets pumped out via a symporter into the lumen of the gastric gland as hydrochloric acid. So we took a weak acid, which is carbonic acid, and we dissociated it into carbonate, and then we exported hydrogen chloride acid. So one of the things here that you'll notice is, did anybody ever run like cross country in high school, or had to do like a lot of running or intervals or something? What happens if you like push yourself too much? You hit the wall. What does that mean? Looking for two words. You barf. You barf. That's what I'm saying. Who's ever barfed because they've been running so hard? I used to run cross country. It happened to me a couple times. You're running and you send your body into acidosis, right? Well, this is one way to get rid of some acid from the body. If you vomit, you're removing hydrochloric acid and alkalizing your blood. This is exactly why you barf when sometimes after really extreme exercise. It helps to stabilize the blood pH. All right. I don't know if that's interesting or not, but now let's talk about chief cells. Chief cells produce this zymogen or proenzyme, meaning an enzyme that is not active. We've heard that word already, zymogen. It's called pepsinogen. Pepsinogen, in the presence of hydrochloric acid from these parietal cells, gets this regulatory peptide cleaved off and becomes peptin. This is a peptidase. Pepsin is a peptidase. What a peptidase does is it takes peptides and it breaks them up into smaller peptides. All right? Pepsin takes these dietary proteins that you've eaten, and it's a protein that breaks down other proteins. So it cleaves these dietary proteins, your steak or tofu, whatever you like to eat, breaks it down into smaller amino acids and smaller peptides. Pepsin. Okay, so that's what the chief cells do. We'll get the G cells in a minute. All right. Meanwhile, let's talk about the phases of gastric secretion. Phase 1, step 1. It's the cephalic phase. I am gonna make big 3.5-pound chunk of salmon. It's been in my fridge for a day. I'm gonna cook it tonight, and I'm gonna cook up a vat of red lentil soup with some turmeric and carrots and celery to go with it as an appetizer. I'm gonna lay out a big spread of all these delicious avocados because they're in season right now. I got these pomelos, these giant grapefruit-looking things. I'm gonna peel that and serve that with the avocado and put some flaxseed oil and ume vinegar over the top of that. Maybe some, yeah. You know what I'm talking about? That's happening tonight. I'm gonna do that tonight. Last lecture. And that soup is gonna smell good. That soup is gonna smell good. all of that, what you were just experiencing is your vagus nerve, the wanderer, the lonely wanderer, looking for someone to listen to it, coming down to the stomach and starting the cephalic phase. The thoughts of food, the smell of the food, maybe you taste a little bit of it. Okay, getting the ball rolling via the vagus nerve. It's going to turn on the mucus cells. Let's start getting some fluid in there. Let's turn on these chief cells to create the pepsinogen. Let's turn on the parietal cells to make some HCl and start activating that pepsinogen. We're ready. We are ready. Let's turn on the G cells. G cells are going to make this enzyme called gastrin. We're going to get to gastrin in just a minute. So all of this is going to start, this is all coming through the submucosal plexus. This is going to start the ball rolling, cephalic phase. Next, oh yeah, the gastric phase. It's time to eat. All right, so our stomach is getting full. We're sending food down. And we're turning on the stretch receptors in our stomach. We're turning on the chemoreceptors in our stomach. The submucosal and the myenteric plexuses are on, and there's all kinds of rumblings going on. The stomach is mixing the food up. And the partly digested peptides that are from this pepsin that has been turned into pepsinogen by the HCl, these peptides start activating G cells even more. And so there's all this gastrin that is getting pumped out. And what does gastrin do? This is it's an endocrine hormone that's going to hit the tissue throughout the stomach, and it's going to stimulate Chi cells to make more pepsinogen, stimulate parietal cells to make more hydrochloric acid, and it's going to stimulate the longitudinal, circular, and oblique muscles of the stomach to start doing this. All right, the gastric phase. Okay, this is dinner. So now we're on to the intestinal phase. Oh, that was a meal. That was so good. It's delicious. I'm full. Thank you. No more. What, peach pie? Oh, God. Maybe. Okay. All right. But after that, I'm done. Thank you. So the food is passing from the stomach through the pylorus, through the pyloric valve, into the duodenum. And when that duodenum gets in contact with what was the stomach contents, we have the production of these different hormones. We have cholecystokinin, we have a gastric inhibitory peptide, we have secretin. So cholecystokinin and gastric inhibitory protein get turned on by the presence of lipids and carbohydrates, respectively. And then secretin is pH. When you have a drop in the pH in the duodenum because you have all this acidic stomach contents get in there, it starts making secretin. When that happens, when we start getting stuff in the duodenum, these three hormones are going to start turning off everything that we had turned on. It's turning off the lights in the stomach as we're moving on. So this is a general motif. So as food moves along, a hormone is going to be made that's going to turn off the mechanism behind it. All right? Does that make sense? So this is the intestinal phase. So hormonal aspect to it, and this is a neuronal aspect. There's these duodenal stretch and chemoreceptors that have this short reflex that helps turn off the myenteric plexus in the stomach. I'm going to sneeze. All right, on to this guy, Barry Marshall. You know this story? Yeah. Does anyone, who's heard this story before about Barry Marshall? Just a few of you? I don't know. It's pretty gnarly. Okay, this guy here, Dr. Barry Marshall, he was a researcher in the late 70s, and he was maybe an MD, PhD, or maybe just an MD, I don't know. but he was researching peptic ulcers. And he started taking tissue scrapes of some biopsies of these peptic ulcers and plating them out. And he found that all of these peptic ulcers, so here's the pylorus, right? The pyloric valve. Here is a peptic ulcer. He found that this really common bacterial infection there. And these bacteria are acid resistant. They can live at pH 1, no problem. Helicobacter pylori. Helicobacter pylori. So he's like, well, golly, I think that ulcers are caused by Helicobacter pylori infections, not by being stressed out and eating corn dogs. You may have gotten them from the corn dogs if you're visiting the intestinal crypt, but it's not every corn dog is going to do it for you. And everyone said, you are crazy. It is clearly because you're at the intestinal crypt. And he said, okay, well, what do I have to do to prove it to you? What would you do? What would a responsible scientist do? Science. They would. A responsible scientist would do science. It's almost a tautology there. What do you think? So this guy got a big flask of Helicobacter pylori swimming around in some chicken broth, and he just downed it. He downed it and gave himself a humdinger whiz-bang case of peptic ulcers, like really bad. And then what did he do? He followed it up with a course of antibiotics and totally cleared it up. Fast forward, 24 years, 25 years, and dude gets a Nobel Prize. So don't let other people who don't believe in you stop you from doing what you think is the right thing. I guess that's the lesson. Is that the right lesson to be? I'm not sure. Don't eat at the intestinal crypt is probably a safer lesson. But if you do, make sure you have some antibiotics on hand. All right, on to the small intestine. So the duodenum is called the mixing bowl of the stomach. It's 10 inches long, and this is where we're going to squirt in the pancreatic juice, squirt in the bile, get those intestinal contents to further break down. All right, we're going to neutralize the acid because a lot of the sugars that are there are going to be broken down by an enzyme called pancreatic amylase, which is like salivary amylase, but it doesn't work at low pH. So we got to neutralize those acids. After the duodenum, we move into the jejunum, and this is where all those acids get to work. They break it down, and then this is where nutrient absorption really begins in the jejunum. And this is about 8 feet long. And then onto the ileum, about 11, 12 feet long, where we continue the absorption. 90% of the food that you absorb as nutrients are absorbed in the small intestine, okay? 90% of the nutrients are absorbed in the small intestine. The large intestine is actually just about compaction and water resorption, all right? Compaction and water resorption. What else? There's a number of reflexes here. The one that opens that ileocecal valve is called the gastroileal reflex. All of these reflexes, as I said, are going to be stimulated by the passage of the food down the chain and stimulating the relevant myenteric plexus throughout the length of the intestine. All right, so we hit the high intestinal surface area drum pretty hard in lab. I'm not going to bang that harder here. There's no point. But we can, just to show this picture with all these, so here are some villi, and you can see all this food stuck around there. And so there's like really high surface area where this food can get absorbed across all of this plasma membrane, all right, of all the cells that are in the villi. This is not the surface of a single cell. This are many, many thousands of cells in this picture, right? Those are projections, the villi. Okay, so then on to the pancreas. The pancreas it's almost like two organs in one. One part of the pancreas is endocrine, and the other part of the pancreas is exocrine. So we've already talked about the difference between those a little bit. Maybe I didn't. I didn't really get a chance to talk about the integument here, did I? Merocrine, apocrine, did I talk about that a little bit? Endocrine is the production of some kind of cell product, usually a hormone or something, that's getting sent into the bloodstream. Whereas exocrine, this is just to remind you, is a cell product that's getting released into the duct of a gland. All right? The duct of a gland. So the pancreas is two organs in one. The first part of it is the pancreatic islets, the islets of Langerhans. There's another good eponym. I'm sure there's a story behind it. I can't tell it to you. Sadly, I will someday. It makes insulin and glucagon. So insulin is required for all of your cells to be able to take up glucose from the blood, and glucagon is required for the cells to begin breaking down glycogen reserves. Right? So insulin and glucagon made by the pancreatic islets, the islets of Langerhans. Then, I'm not going to talk about that anymore right now, but I'm going to move on to the exocrine cells. So the exocrine cells produce what's called pancreatic juice. Oh, I will say this about the pancreatic islets. Just because I had it, I was reminded of it earlier, that type 1 diabetes, juvenile onset diabetes, is an autoimmune disorder that kills off these islet cells and leaves everything else intact. But these people are not able to make, they don't have pancreatic islets and aren't able to produce insulin and glucagon. So the exocrine cells make this pancreatic juice, goes down the pancreatic duct. It's full of this beautiful brew here. We've got alpha amylase, pancreatic amylase. This is going to cleave the disaccharides, the alpha-1,4 glycosidic linkage. It's got a lipase. A lipase is an enzyme that breaks down fats. It's going to convert triglycerides into fatty acids and monoglycerides. We've got proteases and peptidases. These are zymogens, so they need to be activated. We'll see a bunch of them. Trypsin is the big one, trypsinogen. And then there's also ribonucleases and deoxyribonucleases. So these are proteins who are going to break down DNA and RNA that might be in the food that you eat. Okay, so moving on. So I'm going to talk at different points today about the various macronutrients, carbohydrates, lipids, and fats. Here's carbohydrates. So here's maltose. I don't know if I've told you this, but my actual background, the reason I have any qualification for being here is I have a PhD in carbohydrate chemistry. I'm actually a theoretical carbohydrate biochemist. That's or biophysicist, I guess, is what my background actually. So here's a little bit of carbohydrates for you. This is maltose. It's a disaccharide. And it's a disaccharide of glucose on a 1, carbon 4, an alpha, because it's axial, an alpha-1,4 linked disaccharide of glucose to glucose. So glucose alpha-1,4. That is maltose. That's the stuff you'd stick in like a malted milkshake. Has anyone ever had a malted? Yeah, they're great. Malted milkshakes are delicious. I hope you guys all have breakfast. Amylase is going to cleave this and give us glucose, just plain old glucose. Have you ever eaten just plain maltose before? Who's had what are those things called? Huh? Whoppers, thank you. Who's had a Whopper before, that Halloween candy? That's what maltose tastes like. Yeah, they're malted milk balls covered in chocolate. Pretty good, pretty good. Not super, super sweet, they're nice. And has anybody ever had glucose syrup before? Or what's another one? Brown rice syrup. Has anyone ever tasted brown rice syrup? Yeah, brown rice syrup is got no fructose in it. Supposedly better for you. What another barley malt? Barley malt sorghum is another way you would maybe get to taste some glucose. So glucose isn't super sweet either. Anyways, amylose, vegetable starch, is just a long chain of this. It's just a long chain of that stuff with branches, but it's all this stuff. So pancreatic amylase takes this starch that you see up there, the amylose starch, breaks it into disaccharides and polysaccharides that are going to come in contact with different enzymes that are embedded in this so-called brush border. And I used to have more slides about that, but I took there are these enzymes. Maltase is going to cleave maltose into glucose. Dextrinases. Dextrin is D-glucose. Glucoamylase. There's a bunch of these different glycosidases that are going to break the glycosidic bond. And anyways, the whole purpose of this is to get glucose, free glucose that can be absorbed. So your salivary amylase, your grandma, your mom, I don't know, somebody, maybe your dad, I don't know who, but hopefully somebody told you to chew your food well when you were a kid. And the reason you would do that is because salivary amylase is really important in helping to digest the sugars. In fact, half of the starch you eat gets digested before it even reaches the intestine, just in the mouth. All right? And salivary amylase turns off in the stomach because when you get below 4.5 pH, it turns off, it won't work. And so then the pancreatic amylase and these brush border enzymes take over and finish up the job. All right, on to proteins. So there's a protein called trypsinogen. It is a zymogen, an inactive protein that's converted into an enzyme when activated by another enzyme. So trypsinogen is 229 amino acids long. And it's got this little tail in the cartoon I showed as this little green box. There is a brush border enzyme like those other ones, the maltase and the dextrinase, those other enzymes. There's another one called enterokinase. Enterokinase. The job of enterokinase is to cleave off this little thing on the end of trypsinogen and leave behind the 223 amino acid long trypsin. Trypsin is actually active. And trypsin is quite a little buzzsaw of an enzyme. The job of trypsin is to activate all these other proteases and peptidases. In fact, it activates enterokinase, it activates itself, it activates chemotrypsinogen, it activates procarboxypeptidase into chemotrypsin and carboxypeptidase. Yeah, trypsin is the switch that gets tripped and activates all these enzymes. And the job of these enzymes is going to be to break down proteins into amino acids. Okay, so there's this idea of proteins and nitrogen balance. So In a person who's not growing or shrinking, who is healthy, a healthy person, a healthy adult that's not growing or shrinking, the nitrogen that goes in your mouth in a day should equal the nitrogen that leaves your body in a day in amount, right? Molar equivalents. And that nitrogen's leaving your body. Some of it's in your feces. As anyone who has had a dog that likes to eat poop will know, they like to do that because they know there's plenty of protein in your poop still. But a lot of protein is also in your urine. I showed you that yesterday. Well, let's look at this. So your normal person's eating about 80 grams of protein in a day. You lose about 10 grams of that in your feces. The body takes up the rest, and it cycles around into your body protein. Some of it gets broken down into amino acids and the lower metabolites like the urea and the ammonia and the creatinine and all the other stuff that I showed you yesterday. And about 70 grams is going to get excreted in your urine. All right, so this is a person in what's called normal nitrogen balance. So now we have these two situations. We have positive nitrogen balance. This is where the absorption of nitrogen is greater, nitrogen in is greater than nitrogen out. And here are a few examples of where this might happen. A bodybuilder who is actively putting on muscle, all right? Someone who is actively putting on muscle is going to be in a positive nitrogen balance. A woman who's pregnant, who's growing a fetus, that person, their need for nitrogen is high because they're building protein. So they're going to be consuming more nitrogen than they're losing. A child is in positive nitrogen balance because their body is growing. They're building muscle mass. This should be pretty straightforward. So if you were to track the amount of nitrogen that they were consuming, it would be greater than what you would be able to find in their urine and feces. And then on the other hand, there's negative nitrogen balance. This is where the nitrogen in is less than the nitrogen out. Okay, and so some examples of this can be seen in people with anorexia nervosa. So anorexia nervosa, these people are not eating enough calories, and consequently, their body begins to consume protein as a source of energy. It's not the ideal source. But when that happens, they have nitrogenous byproducts of that, and the nitrogen is leaving their body as urea and ammonia much faster than they're consuming any kind of nitrogen. So that's one. Another example is the unfortunate case of starvation. So this same molecular process of the child in the middle. And then as to the woman with anorexia that you see. And then lastly is any kind of wasting disease. So I'm showing Stephen Hawking there with his Lou Gehrig's disease or Lou Gehrig's syndrome. Again, the body he's not burning muscle fiber for fuel. Instead, there's this de-innervation of the muscle, and the muscle's dying. But as the muscle dies, the protein is broken down, and the nitrogen is excreted in the urine as well. So these are examples of positive and negative nitrogen balance. All right. So I did carbohydrates, I did proteins a little bit. Now I'm going to do some fats. I wish I could go so much deeper into these topics, but we'll just get a little flash through it. I don't know how many of you have had how much biochemistry, so I'm not going to assume that you've had a huge amount. Just show you what a fatty acid looks like. A fatty acid. Is anyone familiar with fatty acids, the structure of them? Most of you? Some of you, sort of? Okay, good. Well, a fatty acid is just a long chain of carbon. It's a long chain of carbon. And at the end of the fatty acid is a carboxylic acid. That's it. They're really simple. Just a long carbon chain, fully saturated. A saturated fat would be one where all of the carbons have all hydrogen bonds on them. An unsaturated fat is one where two of the carbons are connected rather than by a single bond, they're connected by a double bond. All right? That's an unsaturated fatty acid. So this carbon here has only one hydrogen attached to it. It has 3 bonds to carbon. Whereas all the other carbons in this chain all have 2 hydrogens coming off. All right? So saturated versus unsaturated fatty acids. If it was less pressed for time, I would go into the biophysical implications of that, but we're not going to. So we can have monounsaturated fats and we can have polyunsaturated fats. A monosaturated fat is a fat that has only one double bond in it, one site of unsaturation, and a polyunsaturated fat can have multiple sites, right? I'm showing one that has 2. When you're thinking about the unsaturated fats, the number and position of this first site of unsaturation is called it's relative to the omega carbon, omega being the last carbon, the last letter of the Greek alphabet. So this would be a 1, 2, 3, 4, 5, 6. This is an omega-6 fatty acid. It's an unsaturated fatty acid. Omega-6s are good. You want to eat them. In general, unsaturated fats are vegetable fats, and saturated fats are animal fats, although that's not completely true. There's plenty of saturated vegetable fats as well, like palm oil, coconut oil, stuff like that. All right, so let's see what this. Let's see some implications of the saturation and unsaturation. Here is a cis fatty acid. It's called cis because the 2 chains are on the same side of that double bond. The thing about double bonds is they can't rotate. You can't rotate around a double bond. They're locked into place. I didn't bring in my molecular models to convince you, but if you take organic chemistry, they'll prove it to you there. The 2 big chunks on the same side of that double bond are called cis, and this is a trans fat. They're on either side. So on opposite sides of that double bond. The thing here is that these naturally occurring cis fatty acids, because they have this kink in them, they don't pack in together very well. They allow more mobility, more flexibility in the membranes that they are embedded in. And because of that, they tend to be oils, not fats. They have a lower melting point. Trans fatty acids are not natural. We make trans fatty acids by heating these bad boys up, these cis fatty acids up, and creating trans fatty acids. Trans fats are fats. They tend to be solid at room temperature. And they're also actually quite bad for you for a number of reasons, which is a discussion that I definitely don't have time for. Did I go past? No. Oh, it's only 11:30. Jeez. For some reason, I had I don't know. Okay, we're cool. It's just like, man, how am I going to get through this? Am I going too fast? Okay, good. I've been watching the clock being like 20 minutes. Oh, my God. All right, no problem. Yeah, trans fatty acids are a problem. So here's some of the problems with trans fats. They lead to coronary artery disease. They lead to atherosclerosis. I always have to say that word slow. I cannot say it fast. Atherosclerosis. Just can't do it fast. Coronary artery disease, myocardial infarction. So let's see what that means. First of all, it's these plaque deposits in the wall of your vessel, all right, and in your heart. So we said that the coronary arteries were the first ones to come off the aorta, right? And because of that, that's where the flow is really intense and kind of turbulent. You're highly likely to have an inflammatory process start in the coronary arteries. And so we get this deposition of fatty plaques. Now that I just gained a half hour in the class, I will certainly be able to show you some slides of how that happens later in the talk here. But when this becomes completely occluded, we get an infarction in the tissue of the heart wall. An infarction is just cell death. The tissues don't have oxygen. They go hypoxic and die. We call it an infarction. Who had the pig that had the infarction? Yeah, you guys. These two had a pig that had like a clear infarction, a darker circle on the wall of the heart. That was pretty interesting. Never seen that one in a pig before. So what's the toll? 600,000 people die of heart disease every year in the United States. That's about 25% of everyone who dies has some kind of heart problem. Heart disease and stroke cost America. This is staggering. This statistic was as of this fall. $1 billion a day. $1 billion a day. You could break that down in like a minute of time and think about how much it's costing America every minute. Trans fats. In medical costs and lost productivity. That's not just something I got off of WebMD. That's a direct CDC stat. So here is the incidence of myocardial infarction and heart disease. This map is as of 2009, but by county in the United States. By county in the United States. What do you notice there?
South.
Yeah, that Paula Deen diet's not really working for them down there. No. And that's because there's a lot of fried food down here, right? A lot of fried food where they're taking vegetable oil and unsaturated fat, they're heating it up to a high temperature, producing trans fats in the process, and then consuming it. Your whatever, the greasy food, the junk food. Fried, all the stuff that tastes good. Hey, what a drag. Actually, carrots taste delicious. All right, so that's trans fats and atherosclerosis. Yes, sir.
So if that's trans fats, how is saturated?
Yeah, that's a good question. It turns out that saturated fats will, if you have a diet that's too high in saturated fats to the exclusion of other fats, that will also lead to atherosclerosis. But saturated fats, the difference here is that saturated fats still can move around. A trans fat at that point is locked in place. It's locked in place, first of all. So there's that. Secondly, I didn't put a slide up that shows why, the origin of the melting point temperature and the effect it has on membrane dynamics as the fats integrate into the lipid bilayer. But essentially, it's the types of hydrophobic interactions that the lipids have as they're packing. Should I go off script and find that slide? No, you don't need it. You want me to? You want me to? Okay. See if I can find it. the packing forces in those lipids. Let's see if I can find it here. Nope. The packing forces, I had it open. Okay, here we go. Oops, yeah, you're going to see that later. Oh, here we go. Yeah. So, oh, this is going to do its thing again. Come on. Yeah, whatever. Well, I guess they're on the same screen. So here are some saturated fats all packed into one another. Here are some cis unsaturated fats, right? And what keeps the membrane together is the hydrophobic interactions, right? It's the hydrophobic interactions. So these have a much higher density of hydrophobic interactions than they have over here. But if you had these, instead of being cis, you had them being trans, they would line up like this. It's true, but the interactions between the pi electron interactions between those the alkene groups are going to have a higher affinity and change that membrane dynamic compared to a saturated fat. So that's one aspect. The other aspect of it is that the steroid hormones that we make in our body, the leukotrienes and whatever, there's all these different steroid hormones that we make out of fats. The type of steroid hormones you make is dependent upon the type of fats you make. So a saturated fat is going to give you a slightly different hormone profile than a trans fat. Does that make sense?
Yeah.
Okay. Yes, sir. Man. The one thing I do have in this slideshow that's kind of fun, I got this slide. I love this one. Wait, I said you're the good kind of fat. I love that. I was talking about avocados in that slide, or the slide before that. They're great. Avocados are great. Avocados are great, the take-home message of the day. All right. Let's make this actually work. All right, so triglycerides. We can take these fatty acids and we're going to pack them. We need a way to package them. And that package is called the triglyceride. It's one of the fundamental units of packing them. There's going to be larger structures that we make out of these, but this is the first way we're going to pack it. And that is basically what's called a dehydration reaction. So we're going to take the alcohol group off of this carboxylic acid on the fatty acid, and the proton, the hydroxyl proton, off of the hydroxyl group on glycerol. So glycerol is a 3-carbon chain that has 3 hydroxyl groups, 3 alcohol groups on each of the carbons. All right? And we're going to dehydrate it. We're going to take water, make water, chuck water off, and then connect this carbon to this oxygen. And we get this ester linkage right here. All right? That's what that's called. So we have a long carbon tail attached via this ester to this 3-carbon fragment. That's a triglyceride. Tri because there's 3 fatty acids attached. Glyceride because it's attached to glycerol. Triglyceride. You guys with me? I know this is more chemistry than you probably thought you were getting into, but you can do it. I'm not going to go too much deeper down the well than this. Okay, so triglycerides. Let's set them aside, and we're going to figure out what to do with triglycerides in a moment. So we need some bile. Bile's made by the liver, and I kind of short shrifted the liver here, but the bile is composed of these bile salts. And the job of a bile salt is to break up the fat molecules, like the globules of fat. Because say you eat something, I have a friend named Aaron that I went to undergrad with. And one time, just because he's out of his mind, he just looks at me and took a stick of butter and just stuck it in his mouth and ate the whole thing. I was just floored. I was floored. It was, I don't know what to say. Anyways, so he had this big glob of fat in his gut at that point. And it was his liver's unfortunate task to try to deal with it, right? And his liver kicked into high gear and started producing all of these bile salts. And there's a number of them, but here's one of them. You don't need to memorize the shape of it or the structure or anything. But it's just to say that it's made out of cholesterol. So if we look at cholesterol, it's this long, greasy body. That's all hydrocarbon, except there's one little hydroxyl group sticking off the end. So it is amphipathic, meaning it's got a polar end and a non-polar end, all right? And it can stick into a membrane. We take cholesterol and we kind of gussy it up a little bit and stick all this other stuff on here, and we make a bile salt, all right? So there's going to be this big hydrophobic side, this non-polar side, the side that does not want to be near water. And there's going to be this side that likes to be near water, polar. It's polar, wants to be near water. All right? So this big fat globule, this baseball-sized lump of butter that my friend Aaron ate, it gets broken up. The bile salts begin to attack it and break it up into all these little fat droplets that have different phospholipids, triglycerides, and the bile salts, packing it in. So there's like a greasy core, and then there's a nice polar shell to it. Okay? Who's with me? Alan's with me, most of you guys. Okay, good. All right. So we've got these fat droplets, and we got to get them into the blood somehow. You know, these triglycerides have like glucose envy here. Here's these glucose, these sugars, and what are they saying? They're hydrophilic. They're saying, come on, triglycerides, let's all jump into the bloodstream. And the triglycerides say, whoa, 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 we can't swim. Don't know how to do that. They're hydrophobic. So we got to get them into the blood somehow. How do we do that? Lipoproteins. And we're going to see what that means here in a bit. Right now, in fact. Okay. What is a lipoprotein? So it's a cluster of cholesterol, triglycerides, other lipids. There's a lot of different categories of lipids. I've just barely scratched the greasy surface. That are surrounded by a hydrophilic. So I've been using this term assuming you guys all know what it means. What does hydrophilic mean? Water-loving. Yeah, water-loving. A hydrophilic protein coat. And we can classify the lipoproteins by their density. So there are 5 categories. There are the ultra-low density, the very low density, the intermediate density, the low density, and then the high density. All right. And the lowest of them, the ultra-low density, are also known as chylomicrons. Because they're made from chyme, or your bile, and they're about a micron in diameter. Or I guess 1,000 microns, a millimeter, yes. All right. So here's how this works. We're gonna take the path of fat through your body. Here's that lump of butter my pal ate. This is the inside of your gut. Here are the villi and the cells, the epithelial cells on the intestinal lumen. Here's a lacteal up in a villi. A lacteal, I pointed out, was that lymphatic thing, that lymphatic structure that connects with the lymphatic system. The lymphatic system connects to your circulatory system via this thing called the thoracic duct, which is like right about here. I should put a picture of it, but it's in your thorax right about here. The thoracic duct is what this is, and it joins with the circulatory system. Here's your liver, here's your peripheral tissues, okay? You got the map, we're gonna step through it. So, first of all, fat glob forms in the intestines after a meal of solid butter or whatever. We form these emulsion droplets as the liver releases the bile salts, and it emulsifies it. That means it breaks it up and gets it suspended into solution. All right. Then that breaks down into even smaller bits called micelles or micelles, which are super teeny tiny little droplets of fat that have these bile salts on the outside of them. Slowly, individual fatty acids are absorbed or leached off of these micelles and brought into the enterocytes, which is the other word for the epithelial cells that's on the lumen of your gut. So the enterocytes absorb the fatty acids, and then there, in these enterocytes, they get packaged into the chylomicrons, into the ULDLs. And they're going to use a bunch of amphipathic proteins and different phospholipids to help create these lipoproteins, the ULDLs, ultra-low density lipoproteins. It's these ULDLs that the lacteals are able to handle, and they go into the lacteals. They pass through the lymphatic system, through the thoracic duct, and enter the bloodstream and head straight for the liver. They make for the liver. The liver is going to release, and I'll give you the short answer here, the good fat, the good cholesterol. It's going to release high-density lipoprotein. High-density lipoprotein is what you want in your body. It's what you want in your blood. All right, it's what's going to help prevent atherosclerosis. So HDL is pretty amazing. This set of slides is simplistic, but I'm going to give you a little bit of it. HDL, one of its jobs is to peel triglycerides and fatty acids off of the chylomicrons and help deliver those to the peripheral tissues. So your tissues need fats, right? They need fats. This is why we eat them. They're there to help maintain plasticity of the membranes, and they do all kinds of things. But the HDL peels the triglycerides and the fatty acids off of the chylomicrons. I call them tags there. Tag for triglyceride because it's the full word would be triacylglyceride, but triglycerides. Acyl just refers to the long chain of carbon at the end. That's an acyl chain, triacylglycerides. Okay, so then HDL also circulates around back towards the liver, and in so doing, monitors whether a cell needs more cholesterol, or if it needs to pick up some cholesterol. Maybe the cell has too much in a peripheral tissue, so the HDL will pick it up. Or maybe it needs to deliver some cholesterol. It'll do that. It tries to get a balance. It's sort of like a homeostatic control of cholesterol levels in the peripheral tissue, depending upon the need of that tissue. So that's HDL. Another function of HDL is once these chylomicrons go to the liver, they get processed there and re-released as VLDLs, very low density lipoprotein. So this is one extreme of the spectrum. This was the other. This is the next step down from the ULDLs. Okay, the next step down, and we're gonna sequentially make this smaller. So HDL does the same thing to VLDLs that it did to chylomicrons. It just keeps peeling off triglycerides and fatty acids. And in so doing, they become intermediate density lipoproteins. Same thing continues to happen. HDL does the same thing to them. And the IDLs continue to get smaller and become LDLs. All right? So the job of an LDL is to just bring the triglycerides and fats that are left over back to the liver. That's the job of the LDL. But sometimes, if there's too much LDL, a macrophage can just say, that looks tasty. And it can come surround the LDL and create what's called a foam cell. It is these foam cells that, with time, cause inflammation and then deposit themselves on the walls of your arteries, leading to atherosclerosis, or hardening of the arteries. All right? So the more fat, the more trans fats you have, the more chylomicrons you have, and then the more VLDLs, and then at the end of it, the more LDL you have, and the more foam cell. So what you want is a high HDL so that you can deliver as much of these triglycerides and fatty acids as possible and minimize excess LDL and foam cell deposits. You want a higher HDL to LDL ratio is better for you. When they run your blood lipids, that's what you're looking for. All right, so that was pretty intense there. We just went through fats. I talked about lipids a little bit. I talked about proteins a little more, and I talked about lipids quite a bit more. So let's review the chemical events of digestion. So in the mouth, we have salivary amylase and lingual lipase. Salivary amylase is going to start breaking those carbs down into disaccharides and trisaccharides. Those are going to move on to the small intestine, where the pancreatic alpha amylase is going to continue to break them down. And then we have these brush border enzymes that are going to make monosaccharides that will get absorbed by the enterocytes and eventually actively transported into the bloodstream. Okay, so that's carbohydrates. Lipids, which I just talked about, we're going to have lingual lipase helps liberate all these monoglycerides and fatty acids into the lumen of the stomach, which we went through that long sequence here that enables them to be absorbed by the enterocytes and packaged into chylomicrons, which go into the lacteals and go through that long process that I just talked about. And then lastly, proteins, you eat them. Pepsinogen in the stomach gets activated into pepsin by the hydrochloric acid, and we get polypeptides, which then make it to the small intestine. And it's these amino acids that are these short peptides that get broken down by dipeptidase, turns them into amino acids, and they get absorbed into the bloodstream in a manner very similar to that of the sugars. So proteins and carbohydrates are fairly similar. Lipids have their own special way of getting absorbed. All right. So the next concept that this will probably be the last big concept that I give you, is the notion of absorptive and post-absorptive states. And I'm going to correlate this notion of absorptive and post-absorptive states with your metabolism and how your cells, the internal respiration of the cells. So I've kind of been working the whole semester. Everything I've been putting together has been leading me to get you to the point of internal respiration. All right? And this is where I'm going to take you to the doorway where physiology meets a biochemistry class, and then I'm not going to walk through it. All right, I'm going to take you up to that door and leave you at that door, and you can take a biochemistry class to go further. But post-absorptive and absorptive states will get us there. So what are they? The absorptive state is the period in which the gastrointestinal tract is full. We just had a meal. And in your body, anabolic processes exceed catabolism. I'll define those in a little bit. But essentially, anabolism is building and catabolism is breaking down. So we have lots of raw materials. We're going to build stuff. And the fuel in the absorptive state is glucose. The post-absorptive state is that time following a meal, the absorption of nutrients, when catabolic processes exceed anabolic processes. And the fuel that we use during the post-absorptive state is glycogen, which is basically animal starch that we made from all the glucose that we had during the absorptive state. We store it as glycogen, and then the other fuel that we can use is fat. All right? Your body doesn't like to use protein as a fuel if it's in neutral nitrogen balance, right? Which is where your body wants to be most of the time. So this is what it looks like, right? Here is glucose, lipids, and amino acids. In the morning, right before class, you're like, oh man, I got to listen to him talk for 4 hours. I better have lunch or breakfast. Hopefully it's breakfast you're having at 8 a.m. And the circulating plasma levels drop until you get to noon, have another lunch, and then dinner. And then by about 10 o'clock, hopefully, your dinner is several hours in the past, and you enter the post-absorptive. This is all post-absorptive right here. And there's these like little small patches of post-absorptive state at lunch and right before lunch and dinner that trigger hunger. Right, the post-absorptive state is going to trigger hunger and cause you to eat. Except when you're asleep. When you're asleep, you're not hungry. You're just running on glycogen. All right, so let's push these ideas forward. Here's an overview of metabolism, the physiologist's concept of cellular respiration. Yeah, we've talked about external respiration. I've talked about cardiovascular system, how we get everything. All of this is getting the fuel to a cell. How is a cell actually going to use it? What is the basic process of a cell running? And that's what this is showing us. So we have these organic molecules that are in your blood somewhere. They get absorbed, and they enter a nutrient pool in the cell. There's these amino acids and lipids and sugars, whatever, in this nutrient pool that's available for different processes in the cell. Some of the components of the nutrient pool can go through anabolism, the building of larger structures, and that can be the maintenance and growth and repair. It can lead to secretion of cellular products. It can be the storage of reserves like glycogen. Anything that gets built, of course, can get taken apart. And if you take it apart, you got to recycle it somehow. The body doesn't really have landfills, so we have to recycle all those molecules. They can re-enter the nutrient pool. Now, from the nutrient pool, another possible path would be the catabolism of those nutrients for energy to drive all this. So we break it down in the mitochondria. And these glucose and fats, what have you, when we break them down, 40% of the energy output from those chemical reactions, those catabolic chemical reactions, 40% of it is captured chemically in ATP. All right, 40% of the energy, the chemical energy that we are releasing through the metabolism in mitochondria is made into ATP. And this ATP drives anabolism, and it drives all this other stuff that the cell needs to do, right? We've talked about many of these. Locomotion, contraction, transport, cytokinesis, endocytosis, exocytosis, membrane, potential dynamics, like all the stuff that needs energy for it to happen is the ATP. 60% of the chemical energy that we release is given off just as random heat. It's randomized thermal energy. All right. So let's fill in the details just a tiny bit more. Just a tiny, I promised you I wasn't going to turn this into biochemistry. This is just going to be still the physiologist's view. So this is this nutrient pool, and instead of just being general nutrient pool, it's breaking into fatty acids, sugars, fats, sugars, and proteins, right? Or fatty acids, glucose, and amino acids. These are the monomers, the single units. We can catabolically build them up into triglycerides or glycogen to store glucose or proteins, or we can break all of them down into small carbon chains. Pyruvate, acetyl-CoA, for example. Those are the names that you'll hear when you take a biochemistry class. They'll become these small carbon chains, which will enter what's called the tricarboxylic acid cycle, and then we'll go through the electron transport chain. Again, this is like the details of these little boxes here. That's biochemistry. But the bottom line is that small carbon comes in, and we need oxygen to come in here too. I'm not showing oxygen coming in, but we need oxygen to come in to burn this small carbon chains. It gives us carbon dioxide, oxidized carbon, and then water is also the output. And we capture that chemical energy that was released by breaking carbon-carbon bonds in ATP. Specifically, we take ADP, so here's adenosine, here's the A. D is diphosphate P, and we add another phosphodiester bond, another phosphate on it, and go from ADP to ATP and add the third one. All right? This is a high energy bond that when we break it, we can release that energy to do different things in the cell. So all of what I just said, I'm going to talk about it in terms of anabolic and catabolic pathways. So catabolic pathways are breaking things down, as I had said, and they're described in all the red arrows that you see here. And anabolic pathways are in blue, and they're building things up. Larger structures are anabolism. So you've probably heard of like anabolic steroids, right? Anabolic steroids taken by bodybuilders and other unwise athletes are steroids that help drive muscle building or cellular building processes. So this right here, going from glucose to pyruvate, or these small carbon chains, pyruvate, acetyl-CoA, these are the small carbon chains I was talking about. This right here is called central metabolism. That's central metabolism. And going from glucose down to pyruvate is glycolysis. Glycolysis. I'm taking glucose and I'm lysing it. Lyse means to cut. So glycolysis, cutting glucose into pyruvate, and gluconeogenesis, glucose, neo, new, genesis, birth or creation. You're creating new glucose from scratch, from pyruvate or pyruvic acid. All right. So this is an overview of the catabolic and anabolic flow that you would see in the cell. Now I'm going to talk about this in terms of absorptive and post-absorptive state. So here's the absorptive state. This is you're in the intestinal phase of digestion, right? You've got a full belly. What kind of processes dominate in the absorptive state? Well, glycolysis. You've got the money, spend it. You got the money, spend it. Break it down. Break that glucose down into pyruvate. Let's make some ATP. Let's send that pyruvate into the mitochondria and burn some more ATP. Oh, here's oxygen. And finally, picture. We've got the glucose. Let's make some ATP. We're gonna break it down. So glycolysis is strongly favored. And that is for the sole purpose of generating ATP. Everything else that's happening is anabolic. Everything else that's happening is anabolic. The only catabolic process is glycolysis, and it's purely for the production of ATP. That ATP is there to drive all of these other anabolic processes. So since we have glucose, we're flush with raw materials, we're going to build stuff. We're going to build up triglycerides. We're going to build fatty acids. We're going to build triglycerides. We're going to store glucose as glycogen. We're going to make amino acids out of pyruvate. We're going to make proteins out of the amino acids. I have some hormones that guide some of this stuff. Like, for example, insulin here and here drives the formation of fat. And insulin also drives the formation of glycogen from glucose. Insulin also drives the conversion of amino acids into proteins. Insulin signaling is, so in my physiology class that I teach in the spring, or I guess it's Andrea Tilden's, but I've been teaching it the last couple of years. In that class, I make them do a project where they talk about AMPK signaling, which I won't even get into, and also insulin signaling. Insulin signaling pathway in the cell is really beautiful and super complex. But it like touches on all of this stuff. All right. So then let's look at the post-absorptive state. Everything is pretty much the opposite of the last step slide I showed you. So again here, do not memorize individual arrows and individual interactions. Get the big picture and you could then derive all of this. The big picture is that during the post-absorptive state, when you're not eating food, when you don't have raw materials, in general, you're going to be breaking things down. There is a general catabolic flow. There's a catabolic flow. The only thing that is anabolic is the opposite of glycolysis. That's gluconeogenesis. We're going to make some more. Glucose under the direction of growth hormone and the glucocorticoids. They need to be there for that to happen. Otherwise, everything is catabolic. It's catabolic. We're breaking triglycerides down to fatty acids, and glycerol, glycogen down to glucose, glucose down to pyruvate, proteins down to amino acids to pyruvate, et cetera, et cetera. So we can take acetyl-CoA. So a liver cell that produces an overabundance of acetyl-CoA, this acetyl-CoA can be converted into what are called ketone bodies. And the thing that's great about a ketone body is that there's a couple things that are great about them. Ketone bodies can be converted back into acetyl-CoA by any cell in your body. So, here are some ketone bodies. These are different. There's variants of ketone bodies. Any cell in your body can take up ketone bodies and go backwards and form acetyl-CoA, and then send that into the citric acid cycle and make more ATP out of it. All right. So the thing here is acetyl-CoA can come from glucose, but it can also come from fatty acids. It can also come from fatty acids. So, has anyone heard of like the bulletproof diet, or like high protein diets, right? Or high fat diets, low carbohydrate diets? Any of these kind of like weird diet hack things, metabolism hack things. What it's trading on this is the ability of your body to take fats and generate energy from them and release ketone bodies, because ketone bodies can pass the blood-brain barrier and feed the brain. So you can live off of ketone bodies. There's like the bulletproof diet is this diet where like 85% of your calories come from fats, and you eat virtually no carbohydrates whatsoever, and about 15% of the diet comes from, when I say carbohydrate, I'm talking simple sugar. You still have fiber in your diet, but and the other 15% is from proteins. Ketone bodies are also present in people. So for those of you who go into the medical profession, you may see an alcoholic in front of you sometime. And you'll notice that the breath of an alcoholic doesn't just smell like Jim Beam. It also smells a little bit like nail polish. It smells a little bit like nail polish. And you can, and this is because they're producing ketone bodies. They're not eating enough. They're not getting enough glucose. You can also see this in diabetics sometimes, people with diabetes, because they're not able to import glucose, even though they have high blood sugar. This is people with type 2 diabetes or even type 1. They have high blood sugar. They're starving to death. Their bodies are starving to death. And so they still need to make energy, and they're burning fat. Even though they have super high blood sugar, they're burning fat. They can't get the blood sugar in to their cells because maybe they don't have insulin at all. If they're type 1 or type 2, the cells are resistant to the effects of insulin. And so they're burning fat and making acetyl-CoA and ketone bodies, which is getting exported. Yeah. The fat diet? Yeah. I don't know, man. I have to say, I've tried it before because I have a friend who got into it. She's a triathlete. It's like, she's hardcore. And so I went and like hung out with her for a couple weeks, a couple summers ago, and did it. It's interesting what happens to you when you do that. I don't know. I'm just going to say that straight up. I mean, because I could spout conventional wisdom for you if you want, which is going to say you would be insane to eat nothing but fat because you are like on the fast track to atherosclerosis. But I don't know if there's been enough longitudinal study to like hold that up. Some of the results that I've seen show people who have great HDL LDL ratios and their triglycerides are fine. So I don't know. I think it's probably a more complicated thing than we know. There are certainly people, Inuits and Athabascans and people like that live on really high fat diets historically, and they seem to thrive that way. So I don't know. I don't know the answer to that. I mean, I can tell you one thing. When you eat that much fat and you don't have much fiber in your diet, your poop turns weird. Definitely weird. Okay. It's probably more than you wanted to hear. Yeah, I'm sorry I can't give you a more satisfying answer. Yeah, ketone bodies. And they have become really sexy, right? The ketone bodies, there are a lot of ketogenic diets out there versus glycemic diets. And, I think the thing here that it points out is that evolutionarily, humans developed this ability to have multiple sources, or before humans probably, somewhere further back in the evolutionary development of our metabolism. Different food sources require different metabolic ways to deal with them and different ways to manage your energy. So it just makes your metabolism more flexible. Yeah. Okay. So I only have a few minutes here. I can either talk about hormonal control or metabolic disorder. Let's talk about this. There's leptin and ghrelin is, in the last 10 years, there's been a lot of talk about leptin and ghrelin. So leptin, and so this is kind of this teeter-totter thing here, this homeostatic system. Leptin is a hormone that suppresses your appetite, and ghrelin is a hormone that increases your appetite. So before you eat, there's more ghrelin. It's being produced by the stomach. This is when you just enter that post-absorptive state right before lunch, like you're all probably in right now. And your bodies are full of ghrelin, and you're hungry. Whereas during the absorptive state, the fat in your body makes this protein called leptin. It's a hormone that affects the brain and induces a state of satiety or fullness. I'm not hungry right now. All right, leptin and ghrelin. So leptin is produced by the fat tissues in the body. Fat, the enemy that is shortening your life. Banished. How? With sanitized tapeworms. I don't know why that's in there, but it's that's a pretty weird advertisement from the teens. Yeah, hey man, a lot of people do all kinds of stuff for dieting. So dieting is not necessarily all it's cracked up to be, I guess was the point I had with that. So here we go. Look at that poor guy. So we said that leptin suppresses appetite, ghrelin increases appetite, and it leads to leptin resistance. All right. So let's see what happens here. If you eat more calories than are required, particularly in a modern American diet where you're eating too much fructose, where you're eating too high, a diet that's too high in refined sugars, that's going to be converted directly into body fat. If you have an increase in body fat, you are going to have an increase in leptin production in the adipose cells. And if you have too much of that, you're going to disrupt the leptin signaling. All right? And if you disrupt the leptin signal, the brain is going to become leptin desensitized, leptin resistant. The brain, it does not want to hear that leptin signal because it's been hearing it too much, right? So it turns it down, and the brain thinks that you're starving, even though you're quite the opposite. And it increases your appetite. It increases your appetite. You get more obese and hungrier. You eat more, gain more fat, body fat, and so on. And you spiral into this hunger cycle because your body is not able to keep the leptin signal that it's getting in check. All right, so I was going to talk about metabolic syndrome. I guess this is my last slide. Metabolic syndrome, what is it? Well, it's this suite of conditions: central obesity, heart disease, atherosclerosis, hypertension, insulin resistance, which leads to diabetes, low HDL, atherogenic dyslipidemia, so, like high triglycerides, and cancer. These are all things that are part of metabolic syndrome. 34% of the population 20 years of age and over meet the criteria for metabolic syndrome. That's 1 in 3 people your age and older. Yeah, that's a CDC statistic, and that's 2006. Those numbers are worse now because the problem's been getting worse. That medical cost in terms of, the direct medical cost of metabolic syndrome, treating everything in this suite, is $10,000 per person every year. Think about that. $10,000 per person in our GDP gets spent on metabolic syndrome. I put all these diseases up all semester here, talked about how much they cost, this and that. Nothing comes close to this. That's $1 trillion per year in the United States alone. $1 trillion. It's just staggering to me. It's 7% of the U.S. GDP, 7% of the largest economy on earth. And this isn't just our problem. I made this point, but it's really here, metabolic syndrome. I've talked about heart disease. I've talked about insulin resistance. These are all part of metabolic syndrome. That's 1.5 billion people worldwide. 1.5 billion people. There's only 8 billion people on the earth. That's pretty significant if you ask me. That's a fifth of the world's population. 1 in 5 people on earth are plagued by this problem. This is the absolute definition of a pandemic. This is the absolute definition of a global pandemic right here. All right.